Senthil. We used to be together in Japan. I always tell him that though I am from Tamil Nadu, I never studied in TNAU. I missed studying in TNAU. But then when I was doing my PhD in IRI, but then I had so many friends from TNAU and I was so impressed with the quality of students coming out of TNAU. So I'm deeply honored to be amongst you, and I would like to share with you some of my experiences working in CIMMYT on molecular breeding. Okay, of this I will do it especially in the context of maize breeding. Okay, not because other crops are not important, but this is something that I know. So maize breeding is something I am familiar with. Okay. Okay, as many of you might have realized, many of the fundamental concepts in plant breeding, as a student of genetics and plant breeding, you might have realized many of the fundamental concepts have evolved from maize breeding. Right? Be it mass selection, reciprocal recurrent selection, half SIP selection, full SIP selection. So all those basic fundamental concepts of plant breeding have evolved from maize breeding actually. Okay, so the only thing constant in maize breeding is change. So every 2 to 3 years, new concepts keep emerging, and a maize researcher has to keep himself updated on these developments. And my professor in IRI, he always used to tell me, he always used to ask me, do you know what is the biggest limitation in maize breeding? As any student, I could also blink. I would say, I don't know, sir. He will tell me that imagination is the only limitation in maize breeding. So the extent to it, you can imagine, you can design experiments. You know why? Because the maize plant, those of you who have worked in maize, you would have realized, the maize plant, the male and female parts are nicely separated on the plant. It's a monoecious plant. And these things are, the male and female parts are separated, so you can do selfing, you can do crossing, you can do sieving, you can do all kinds of pollination with relative ease. Right? So that's why maize is a wonderful research-oriented crop, and it's also a kind of model species. So okay, so today what we will do is, I will try to give you a very quick overview of what is molecular breeding. So it's also known by many different names, like genomics assisted breeding, marker assisted selection. So all of them are basically same. All of them are nothing but using molecular markers to aid the conventional breeding process. So I will try to give you a very brief overview of what molecular breeding is and how it is changing the way plant breeding is actually being practiced in the 21st century. It's not anymore what we have read, plant breeding in the textbooks. Now things are very gradually changing. So we will just have a look at that. I know that post-lunch session is the most challenging session, right? Because when your stomach is full, your brain is dead. So let us fight today, okay? So keep yourself awake. Keep asking questions. Please don't feel shy. If you don't understand anything, please feel free to ask. So I will try my best to keep you awake. Okay? Just a few words about the institution that I come from. Okay? I come from an institution. It's called CIMMYT. It's a Spanish acronym for International Maize and Wheat Improvement Center. Okay? So this is headquartered here in Mexico. The headquarters of CIMMYT is in Mexico, but it has presence. It has good presence throughout the world. Okay, so we have some collaboration in the northern American continent with Cornell University, and we have a regional center in South America, in Colombia, and in Africa, towards East and South Africa, CIMMYT has good presence. Kenya, Ethiopia, and Zimbabwe. These are the 3 centers where CIMMYT has a presence. And needless to mention, in Asia, it has an excellent presence. I come from, I am based here in Hyderabad. I used to be based in Mexico earlier for around 5 years. Now I got relocated as CIMMYT Asia is expanding now. So there is increasing emphasis on Asian locations. So other than India, we have a presence in China, Bangladesh, Nepal, Iran, Afghanistan, Pakistan, and all those countries. Okay, so CIMMYT has 145 researchers almost throughout these centers. And CIMMYT's mission is to fight world hunger and poverty using advanced scientific tools. And not only scientific tools, but also most importantly, partnerships with local organizations. It can be state agriculture universities like TNAU or other ICAR research organizations or all these national research organizations. And one of the key strengths of CIMMYT is its phenotyping, not the molecular breeding program. The molecular breeding program in CIMMYT is very recent, maybe not more than 10-15 years old. But then one of the real strengths of CIMMYT is phenotyping, its ability to bring about distinct phenotypes in your germ process. So for instance, drought, low nitrogen tolerance, waterlogging, heat. So for all these abiotic stress tolerance traits, CIMMYT has developed excellent sites. Okay. For instance, in this picture you can see that tolerant genotype is well distinguished from a susceptible genotype. Similarly, this is tolerant, this is susceptible, waterlogging. So on this side you see a dark green leaves, right? So that's an optimal fertilized plot, whereas this one is a low nitrogen plot. Nitrogen has been depleted from the plot. Okay. And also similarly in the heat, when it is stressed to heat, you can see the tassel blasting. That's a typical symptom of heat susceptibility. So developing and identifying and developing such environments wherein you can very clearly distinguish the tolerant ones from susceptible ones is the key. And that is really the foundation for the molecular breeding program in CIMMYT. So you should not be under the misconception that molecular breeding means just working in the lab. So I was talking to Professor Maheshwar in the coffee break. I was a little sad. He told me that nowadays students tend to think that they want to be in the lab because they are doing molecular breeding or biotechnology work. They don't want to go to the field. Okay? So but that's not the thing. Molecular breeding, phenotyping is an integral component of molecular breeding. So unless you have a sound phenotyping effort, you can't succeed in molecular breeding. Right? And this is how the CIMMYT abiotic stress tolerance sites are located and distributed throughout the globe. So you have in Mexico for drought and heat, in South America for acidity, for drought again in Zimbabwe and 2 sites in Zimbabwe, and in Hyderabad we have excellent facilities for drought, waterlogging as well as heat. Okay, so that's in brief about the institution that I come from. What I would like to do in the next 30 or 40 minutes, I will try to cover the following grounds. Okay. So there has been a tremendous explosion of genomic information. Whether it's good or bad is up to us to decide. And why I'm saying bad is because the volume of information is growing very rapidly beyond our ability to handle. Right? Because of our computational infrastructure, because of our manpower limitations, we are not able to handle that much of data. That's I'll tell you later about that. And also, how is this developments, the molecular developments, are redefining the way we do maize breeding. Okay. And I will just mention in brief what is the molecular breeding approach that we follow in the global maize program of CIMMYT, and what are the emerging tools. If someone asks you, what are the two major tools that has changed the way maize breeding is done in the last 2 decades? These are the 2 things. One is, anyone has an idea about what is DH? Double haploids. So that has really changed the way maize breeding is done. So if you ask me, many of the multinational private sector companies like Monsanto, Pioneer, Syngenta, 99% of their maize breeding is now based on double haploids. So they have done away with conventional method of developing inbred lines. Because of the ability to produce very large number of inbred lines in a very quick time frame, DH is the most favored approach now. And also molecular markers for a range of traits, not only for simply inherited traits, even for complex traits. Other kind of marker-based approaches are coming out. Kind of whole genome, genomic selection approaches are now coming out for maize breeding. So GWAS, anyone who has an idea of GWAS? Excellent. Yeah, genome-wide association studies or association mapping, what we call, right? And also genomic selection. Genomic selection is more close to plant breeding methods, whereas GWAS is more towards identifying specific genomic regions for a given target trait. And also another important tool called cDNA based genotyping. That's also coming up as an important tool. And what has not improved much, especially in the developing country context like India, is bioinformatics. We don't have enough infrastructure to handle this much data. So a lot of raw data remains raw data. It's not converted into meaningful information. So that's really the bottleneck, and we need to invest really on this, on bioinformatics side. Okay, so before we go on to do that, I just wanted to very briefly bring all of you up to that common table, just to give you an understanding of what is molecular breeding. How is it different from conventional plant breeding? So it's very close to conventional plant breeding because all of these steps, creation, variation, selection, evaluation, release, multiplication, all of these are common steps, right, in any plant breeding program. So what is the difference? What is the single most important difference between molecular breeding and conventional breeding? The second one, selection based on markers. So how would you do selection in a conventional breeding program? You would do based on phenotype, right? So it can be for any observable characteristics. You can do selection based on primate, or you can do selection based on disease reactions, or you can do selection based on biochemical qualities, anything. Whereas in this case, we can do selection based on DNA markers which are linked to the trait. So this doesn't mean that we don't need phenotypes. So before we get to this step, there is an upstream research process here in this molecular breeding context, which is called discovery phase. Right? The discovery phase of the molecular breeding program invests on identifying those crucial linkages between the molecular marker and the gene of your interest. So that has to be done through extensive research. Only then you can get to this step. But then the difference is selection based on markers instead of phenotypes. Okay, so what is that genomic explosion I was talking about? Way back 15 years back when I was a master's student, these marker systems were very popular. RFLPs, restriction fragment and particle physics. By the time I got to my PhD in IRL, SSRs and graphics were very, very popular at that time. SSRs were very popular because they were PCR based, right? But now if you ask me, how many labs really run SSRs? Very few. Now the most important dominant marker system is SNPs, single nucleotide polymorphisms. Why? Because they are abundant in nature, they are predominant, and they are very frequently polymorphic in any germ process. And then the SNP genotyping technology is also very rapidly evolving. Every 3 months to 6 months, you see a new genotyping platform in the market. So that's why, in fact, 95% of our genotyping in CIMMYT is outsourced. We decided not to establish any genotyping infrastructure. Rather, we will make use of private companies providing this kind of service. So we just extract DNA, we send it to a company in UK called K Biosciences for certain marker genotyping, and we also send it to Cornell for high density genotyping through another system called GBS, genotyping by sequencing. So 95% of our genotyping is outsourced. And this is now one of the very, very powerful genotyping platforms. Have you heard about GBS? It's called genotyping by sequencing, and this is invented in Ed Buckler's lab in Cornell, and it's now applied on a variety of crops. And maize, yes, it's currently right now it is churning out close to 2 million markers per sample. Okay, so you can imagine earlier we used to run 100 to 200 SSRs on one sample, right? And that used to take some 6 months to 1 year. Whereas now you are getting in 1 month time. After you send the sample, within 4 weeks, they return you 2 million data points per sample. So that is the explosion I am talking about. So you have tremendous volume of information, and now even beyond going this, now full genome resequencing technologies are also coming into picture. The entire DNA of any variety that you give, it can be resequenced completely. So that means billions of databases, billions of basis information. I am sure this will not stop here, right? So now many other new terms are being coined: epigenome, methylome, proteome, metabolome. So many all these things are going to come up, and it's all going to add to your information database. So out of curiosity, we were just checking in CIMMYT. From the GMP stands for Global Maize Program in CIMMYT. From the start of the biotechnology program in CIMMYT until 2009, how many data points CIMMYT generated through all the fingerprinting or other SSR based applications? Using SSRs, rapids and RFLPs, around 500,000, 5 lakhs data points in one period. Whereas if you see in the last 2010, 11, 12, in 3 years, using this SNP technology, around 5.5 billion data points have been generated, just last in the 3 years. So the most important thing here is, it's nice to say that we generated this many data points, but what did we do with these data points? That is the important question, right? Actually, if you ask me frankly, we are still struggling to find a way of how to make use of this information, because there is information beyond variability. And say if you send 1,000 lines to Cornell, they return you a file which has 1,000 rows and 2 million columns. Right? That is the data matrix that you are looking at. So if it is Excel, if you use Excel, Excel has only 16,000 columns. Right? Whereas this is like 2 million columns. So the most important point is, how do we convert these data points into selection units that we can make use in the breeding program? So how is all of that taking place? How are we making use of this information? You have high density genotypes on one hand, that is coming from Cornell or from other genotyping service providers, and you have very high precision phenotypes, which is the parallel development is going on phenotyping effort, which we are not discussing today so much, but then impressive developments are taking place here. So high quality phenotypes, genotypes, they are combined using powerful statistics. I mean, we can't just rely on the T test or F test that we used to do earlier. So now we need to have automated, sophisticated statistical models that can take care of such high volume information. And also you need high efficiency computing. Many of these data points cannot be analyzed on our laptops. You need some Linux servers or some high efficiency computing systems with at least 32 GB or 64 GB of RAM. I'm not talking about the hard disk capacity, I'm talking about the RAM capacity. So most of the computers we use are just 4 GB RAM or 2 GB RAM or maximum 8 GB RAM. Now 16 GB is coming. But we need 32 or 64 MB RAM to analyze such GB RAM to analyze such data. So all of them basically channel into two different applications, at least in CIMMYT. So one is GWAS, Genome Association Studies, where the objective is to identify specific genomic regions which has a significant effect on your target trait. Your target trait can be nutritional quality, or can be abiotic stress tolerance or biotic stress tolerance. So they all go through this one. Okay? And parallelly the other effort is genomic selection. Right? So this is more genetics oriented, GWAS, where you want to understand how many genes are governing my trait and how much each gene is contributing to my trait. Here, I am not bothered so much how many genes are involved in the genetic architecture of my trait, but I just want to make use of the marker information to identify the good individuals. I want to discriminate the good ones from bad ones. Are you familiar with genomic selection as a concept? So it's very close to breeding program. Maybe I can give you a very crude analogy. Okay? Suppose if there are 100 students, okay, who are willing to get admission into a PhD plant breeding program, but there are only 10 seats. Okay, how do we evaluate? We ask them to do an entrance examination. Right? You write an entrance examination, and then 10 students are selected. They go over as top score. The top 10 get an admission. Suppose, just for a hypothetical reason, just imagine, it's not true, but just imagine that I have a method